Uh, Next part of the lesson is Megaspore Genesis. In previous classes, we have seen the microsporogenesis. The microsporogenesis means formation of microspores. The microspores or pollen grains are formed from diploid pollen mother cell or pollen mother cell by undergoing meiosis. We call microspore or pollen grains. The pollen grains are always haploid. Okay. Now the megasporogenesis. As the name indicates, mega means big. Spore is big. Genesis, formation of birth of megaspore. What is megasporogenesis? A very important question for 1 mark. The process of formation of megaspores from megaspore mother cell by meiosis is called megasporogenesis. This process of megasporogenesis always takes place in the ovule. Ovule means in the nucellus or the megasporangium. Nucellus is a diploid tissue. Okay. Now, we shall see how the megasporogenesis takes place. During the process of megasporogenesis, a deep-seated cell in the nucellus or megasporangium differentiate itself into an archisporial cell. Differentiate means how it develops, then cytoplasm with a conspicuous nucleus or distinct nucleus. Such a cell is called archisporial cell. In brief, in the nucellus, archisporial cell is differentiated. So this archisporial cell divides periclinally and form 2 cells. The outer cell is called parietal cell. The inner cell is called sporogenous cell. Outer parietal cell forms the outer protective layer. The inner sporogenous cell function as megaspore mother cell. Remember, megaspore mother cell is always diploid. That is 2n. It consists of 2 sets of chromosomes. It is just like pollen mother cell, PMC. MMC, megaspore mother cell. PMC, pollen mother cell. Both of them are diploid. Now, the process of megasporogenesis. Explain for 3 marks. The nucleus of the megaspore mother cell undergoes meiosis or reduction division and form 4 haploid megaspores. 4 spores are formed, which are haploid. Okay. The 4 megaspores are arranged in such a way, one above the other, we call linear tetrad. You just refer the diagram. When megaspore divide meiotically, that is first meiotic division, 2 cells are formed. This stage we call dyad. When second meiosis takes place, 4 daughter cells are formed. These are haploid and arranged in linear tetrad. Note, very important point, among 4 megaspores, upper 3 degenerate or disintegrate or become non-functional. The lower cell, haploid cell, only become functional and develop into female gametophyte or embryo sac. Okay. So refer the diagram. You can see in the first diagram MMC, second diagram dyad, third diagram linear tetrad. The development of embryo sac in some angiosperms takes place from single haploid megaspore. Hence it is called monosporic type of embryo sac development. Okay, so that's all about megasporogenesis. Explain for 3 marks. Okay, differentiate microsporogenesis, megasporogenesis is for 5 marks. That's also a very important question. You make some differences, 5, 6 differences. Next is the development of female gametophyte or embryo sac. It is for 5 marks. Explain the development of female gametophyte. Okay. In majority of angiosperms, after the megasporogenesis, 4 haploid megaspores are formed. Among 3 will degenerated. Only 1 become functional megaspore. It develops into female gametophyte or embryo sac. Next, the development of embryo sac takes place from single cell. Hence it is called monosporic type of embryo sac development. A good question for 1 mark. What is monosporic type of embryo sac development? It is common in polygonum plant. Polygonum. Hence it is called polygonum type of embryo sac development. It's not mentioned in your NCERT. Okay. Note, megaspore mother cell is diploid, that is MMC, whereas embryo sac is haploid, N. Now, we shall study the development, important for 5 mark. The haploid megaspore during the development enlarge itself and assume spindle shape. The nucleus, that is haploid nucleus of the functional megaspore, divide mitotically and form 2 nuclei. These 2 nuclei move towards opposite poles and thus form 2 nucleate embryo sac or female gametophyte. It is the first mitotic division. Next, the 2 nuclei of embryo sac again divide mitotically, result into 4 nucleate embryo sac. This is the second mitotic division. Refer the diagram. 2 nuclei. First one diagram is first diagram is 2 nucleate embryo sac. Second is 4 nucleate embryo sac. Okay. Further, 4 nuclei of embryo sac divide mitotically. It means one more mitotic division takes place and form 8 nucleate embryo sac. Third mitotic division. Okay. The 8 nuclei are free nuclei. Free nuclei means not surrounded by the cytoplasm. Because the division of nuclei is always free nuclear. It means the division of nucleus is not followed by the division of cytoplasm. Such a division is called free nuclear division. Okay. 8 free nuclei are formed. 4 at each pole. Okay. Now, the 8 nuclei of embryo sac at maturity organized into 7 celled, 8 nucleate female gametophyte by cytoplasmic division. Okay. Very important for NEET. You can see the diagram. 3 cells at micropylar end form egg apparatus. 3 cells at chelosal end form antipodals. The big central cell contain diploid secondary nucleus or definitive nucleus, which is formed by the fusion of 2 haploid nuclei. Okay. So that is the development. Refer the diagram, different stages. So at the center of the central cell there is 2 nuclei. One nucleus from one pole migrate to the center, other nucleus from other pole migrate to the center. Thus 2 polar nuclei are there, N plus N. Okay. First diagram, 2 nucleate embryo sac. Second diagram, 4 nucleate. Third diagram, 8 nucleate embryo sac. Fifth diagram, organized embryo sac or mature embryo sac. So that's all the development. It is for 5 marks. Okay. Right. Next. Structure of mature embryo sac or female gametophyte. It is for 5 marks. Once again, one more question. Explain the structure of mature embryo sac. Okay. A typical angiosperm embryo sac at maturity shows 8 nuclei and 7 cells. A very important need point or CT point. It is also called female gametophyte. Embryo sac is also called female gametophyte. Okay. Now the structure. The female gametophyte is spindle shaped and haploid. A very important notable point. It shows egg apparatus, antipodals, a central cell with secondary or definitive diploid nucleus. Now we shall explain one by one. Egg apparatus. 3 cells are grouped together at the micropylar end of the ovule and form the egg apparatus. That is, the egg apparatus is always present at the micropylar end of the ovule. It consists of 2 synergids on either side of central egg cell. The egg cell contains the female gamete, the egg or the ovum, which is always haploid. Okay. Now, very important need point. The synergids contain special cytoplasmic strands called filiform apparatus, which guide the pollen tube to enter into the synergid or embryo sac at the time of fertilization. That's all egg apparatus. For 2 marks, explain egg apparatus. Next, antipodals. 3 cells at the callosal end or at the base are called antipodals, which are also haploid. Okay. Then there is a central cell, a big or large cell. The central cell at its center contains 2 haploid polar nuclei, which later on fuse and form secondary nucleus or definitive nucleus at the time of fertilization. So that's all about structure of embryo sac. Egg apparatus, antipodals and the central cell. Now for NEET or CET, very important notable points are there. You just note down. Ploidy, with respect to ploidy. Nucellus is always diploid tissue. That is to N. The egg cell is always haploid because it is formed after meiosis. Haploid, N. Synergid is also haploid, N. The antipodals, three antipodals are also haploid, N. Embryo sac itself is formed after meiosis, hence it's also haploid. Thank you.